Welcome back to ILS 6, and in this submodule we're going to go over the characteristics of the sympathetic nervous system and the parasympathetic nervous system, the secretion of acetylcholine or norepinephrine. Alright folks, let's get started. Neurotransmitters are very small organic compounds that have to meet certain criteria. They're synthesized by neurons and they're stored inside synaptic vesicles, and they can bind to specific receptors in a target cell. After they bind, they're going to trigger a physiological response. Neurotransmitters are classified based on the chemical structure. In the ANS, we're going to focus on the acetylcholine and also the biogenic amines, which are the dopamine, epinephrine, norepinephrine. In the ANS, it only utilizes epinephrine, norepinephrine. The receptors that bind acetylcholine are called cholinergic, and there are two classes. There's one called nicotinic, and there's one called muscarinic. Nicotinic receptors are considered ionotropic, which means that when the acetylcholine binds to this receptor, it's going to open an ion channel and begin the production of a signal called EPSP. EPSP stands for excitatory signal, which is very much like a light switch when you turn on the switch to turn power on the light. In the muscarinic acetylcholine receptor, these are called metabotropic, and these require the help of a second messenger and a G protein to activate a signal that could be either an EPSP, just like the nicotinic, or an IPSP, which stands for inhibitory signals. The adrenergic receptors that bind epinephrine and norepinephrine are going to be in the metabotropic category that also uses G proteins. Cholinergic and adrenergic fibers. In the ANS, it utilizes acetylcholine, ACH for short, and norepinephrine, NE for short, and either one of these neurotransmitters can cause excitation or stimulation, and it can also cause inhibition depending on the postsynaptic receptor and where that target cell is located. The cells that release acetylcholine are considered cholinergic neurons, and there are 4 places where you can find them. Number 1, you can find them in all the preganglionic neurons. They release acetylcholine. They are also found in all the parasympathetic postganglionic neurons. And the exception to this would be in the sympathetic division, but only inside the neurons that innervate the sweat glands, and then the neurons that innervate the blood vessels and some of the skeletal muscle. Those are the only 2 places where you're going to have a sympathetic division relying on acetylcholine. Cells that then release norepinephrine or epinephrine are considered adrenergic. On the right-hand side, you can see 2 divisions, sympathetic division versus parasympathetic division. The sympathetic division has a short pre and a long post. Whereas in the parasympathetic division, we have a long pre and a short post. In both cases, you can see the chemical synapse region. It relies on the acetylcholine neurotransmitter, shown in that red circle, and it binds to a receptor on the postsynaptic membrane to continue the signal. In the sympathetic division, the postsynaptic neuron can release norepinephrine or epinephrine, and it can bind to a number of receptors, alpha 1, alpha 2, beta 1, beta 2, and beta 3. And then for the parasympathetic division, it can bind to different types of the muscarinic receptors. There are 5 subcategories that we'll talk about later in this module: M1, M2, M3, M4, and M5. Synthesis of acetylcholine is destruction after secretion and its duration of action. Acetylcholine is synthesized by an enzyme called choline acetyltransferase, also known as CHAT, located inside the terminal endings of the axon, as well as varicosities inside cholinergic nerve fibers. Varicosities are the swellings where it pockets the nerve vesicles and releases neurotransmitters. In order to regulate this neurotransmitter, we have another enzyme called acetylcholine esterase, ACHE for short, which will split the acetylcholine into acetate ions and choline that is bound to collagen and the glycosaminoglycans. The choline can be transported back at the terminal nerve ending by these high-affinity choline transporters, and you can see here on the right-hand side, the acetylcholine shown in purple gets broken down after it interacts with acetylcholine esterase into acetate and choline. The choline can get recycled back up for reusage, and about 35 to 50% of the choline gets reabsorbed back in for recycle. Synthesis of norepinephrine, its removal, and its duration of action. Synthesis of norepinephrine begins in the nerve terminal and it requires a series of enzymes and precursors to generate this chemical. It starts off from the aromatic amino acid phenylalanine, and then it converts into tyrosine. Tyrosine goes into dopa. Dopa goes into dopamine, and then dopamine becomes norepinephrine. It does not get converted without the help of a series of enzymes. Tyrosine hydroxylase will then attach a hydroxyl group onto tyrosine to make dopa. Then we have dopa decarboxylase, which is also known as aromatic amino acid decarboxylase enzyme. And this enzyme will strip the CO2 off of a carbon and create dopamine. In order for dopamine to be converted into norepinephrine, it requires a specific channel transporter called a VMAT. These are called the vesicular monoamine transporter that is located inside the axon terminal that will then convert dopamine into norepinephrine when it gets back in. In the adrenal medulla, this reaction goes one step further. The norepinephrine can be converted into epinephrine, the hormone inside the adrenal medulla. Roughly 80% of norepinephrine gets converted to epinephrine in the adrenal medulla. That catalyzes this reaction is called phenylethanolamine N-methyltransferase, PNMT for short. And this enzyme will attach a methane group onto that nitrogenous side chain. When the norepinephrine epinephrine is released, it is going to start to decay over a series of minutes, and there are three ways by which the removal occurs. Number one, it can be actively transported at the nerve ending back into the neuron. Roughly about 50 to 80% of norepinephrine gets recycled back up. Some of that will diffuse into the blood and then make its way to certain organs, like the liver. And alongside the tissues, like the enzymes, the tissue will contain enzymes like MAO, which is monoamine oxidase, as well as the catechol O-methyltransferase enzyme, which can break those down for degradation.